All right, here's the basic problem. You're given a DNA molecule, double-stranded DNA molecule, and you're asked to identify the RNA that would result from the transcription of this molecule, and then asked to identify the protein, the polypeptide, that would result from the translation of that mRNA. Importantly, the problem tells you that the lower strand is the template strand. That is the strand that will act as a template for the transcription of RNA. You're also given a genetic code. Needless to say that a problem like this is impossible without the genetic code. You don't need to memorize the genetic code. All right, so how do we approach a problem like this? All right, so number one thing is lower strand is the template strand. So I'm just going to go ahead and copy that. Before I go any further, let me point out to you that I've put this problem in a constant width font. Okay, Courier is an example of a constant width font. Most of the fonts that we use, Arial, Times New Roman, those are scalable fonts. That is, okay, so constant width means that an A is the same width as a C or a G or a T. Okay, that's going to keep everything in register. In fact, a lowercase I is the same width as a capital M. So it's not the most efficient font for reading, but it's very helpful in a problem like this. Okay, so there's the template strand. So what am I going to do? I'm going to transcribe it. And to have that transcript stand out, I'm going to do it in blue. All right, so note that the template is running 3' to 5' left to right. So the transcribed RNA is going to be 5' to 3' left to right. So now I'm just going to go ahead. T base pairs with A, C base pairs with G, G base pairs with C, and then A base pairs with T in DNA, but in RNA, A base pairs with U. And I'm going to be careful about this because I don't want to make any typos. There we have the RNA that results. Now I went ahead and typed it in and I did the base pairing in my head, but there's a simpler way. Note that this RNA is identical in sequence to the non-template strand. All right, so the quicker way to do it would be just to go ahead and copy this non-template strand again, paste it in here, and then change all the T to U. All right, so wherever there's a T up here, there's a U up here. And hopefully I've gotten it right. All right, so now we have the RNA. Now the next step is to go ahead and translate that RNA. So I'm going to go ahead and copy it. And paste it. Now I'm going to make my life a lot easier. All right, so when RNA gets translated to DNA, it starts at a start codon. The start codon is in the genetic code, but it's one that's worth memorizing. It's AUG. So I'm going to go ahead and identify that AUG, and then I'm going to break things into codon. Believe me, this makes your life a lot easier. All right, and after that codon, I'm going to stop, and you'll see why in a few minutes. Now I can go ahead and translate, and once again, to highlight differences, I'm going to change the color of the font once again. So AUG gets translated into methionine, or MET for short, or just an M. UUA encodes leucine, LEU, or just L for short. AAC encodes asparagine, ASN for short, or just N. UAC encodes tyrosine, TYR for short, or just Y. CAG encodes histidine, HIS for short, or just H. And then UGA is a stop codon. All right, so translation ends there. You don't actually have to write in stop. You could include an asterisk or just not have anything at all. But the point is translation stops. The rest of this sequence does not get translated. All right, so that's the answer to the translation product. MET, LEU, asparagine, tyrosine, histidine. All right, so let's go on to the next part, which asks you to consider a mutation. So let me just copy this whole thing over. All All right, so it's the next part says that this T is mutated to an A. So what I want to show you is that mutations are double-stranded, right? So a mutation in one base implies a mutation in the other base. And once again, I'm going to highlight this by changing color. So now the mutated base is in purple, and what it's saying, what the question is asking, is what happens if that T is mutated to an A. Note that if one base is mutated, it implies that the other one is also mutated. All right, because it's the base pair that's mutated. All right, so that's the mutated DNA sequence. All right, so now we just have to go and find out where that T was in the original sequence, right here. Remember, in RNA it's a U, and that now becomes A. All right, so this AUG UUA becomes AUA. Once again, that's in purple. And now this leucine, where UUA encoded leucine, AAU encodes isoleucine, I-L-E. All right, so that's the solution to the second part of the problem. Now for the third part of the problem, I'm going to go back to the same thing again. I'm going to go back to this original sequence. Okay, so the question is asking what if that original sequence were mutated again? So I'm going back to this original sequence. Mutations are not accumulating. And it's asking what if this C were mutated to A? And once again, it implies that both strands of the DNA are being mutated. So this CG base pair gets mutated to an AT base pair. All right, so, and once again, I'm going to highlight these in purple. That C gets mutated to an A. This G is mutated, therefore, to a T. All right, now the lower strand serves as a template, and where that lower strand, where the RNA polymerase comes across this T in the template, it's going to incorporate an A. So that's going to be right here. All right, so that's going to be an A. All right, so now this codon here, which was UAC, now becomes UAA. And where UAC encoded for a tyrosine, UAA is a stop codon. And once again, a stop codon is exactly that. It means translation stops. So you're going to translate these first three amino acids, then the rest of this protein simply does not get translated. Okay, and there's your solution to the third part of the problem.